Today we will learn about lymphoid system. The cells involved in the immune response are organized into tissues and organs in order to perform their functions most effectively. These structures are collectively referred to as the lymphoid system. The lymphoid system comprises lymphocytes, accessory cells, and in some tissues, epithelial cells. It is arranged into either discretely encapsulated organs or accumulations of diffuse lymphoid tissue. The major lymphoid organs and tissue are classified into either primary or secondary, and in essence, lymphocytes are produced in primary lymphoid organs and function within the secondary lymphoid organs and tissues. Now let's study about primary lymphoid organs. Thymus and bone marrow are primary lymphoid organs. They are sites of maturation for T and B cells respectively. Here lymphocytes differentiate from lymphoid stem cells, proliferate and mature into functional cells. In mammals, T cells mature in the thymus, B lymphocytes in the fetal liver and bone marrow. Birds have a specialized site of B cell generation, the bursa of Fabricius. It is in primary lymphoid organs that lymphocytes acquire their repertoire of specific antigen receptors in order to cope with antigenic challenges that individuals receive during their lifespan. Now let's study about development of T cells. T cell development takes place in the thymus. The thymus in mammals is a bilobed organ located in the thoracic cavity overlying the heart and major blood vessels. Each lobe is organized into lobules separated from each other by connective tissue trabeculae. Within each lobule, within each lobule, the lymphoid cells are arranged into an outer cortex and an inner medulla. The tightly packed cortex contains the majority of relatively immature proliferating thymocytes. More mature cells are found in the medulla, implying a differentiation gradient from cortex to medulla. There is a network of epithelial cells throughout the lobules which plays a role in the differentiation process from bone marrow derived pre-thymic cells to mature T lymphocytes. In this model, pre-thymic T cells are attracted to and enter the thymic rudiment. They proliferate below the subcapsular region as large lymphoblasts, which replicate and give rise to a pool of cells entering the differentiation pathway. Many of these cells are associated with epithelial thymic nerve cells, although the significance of this interaction is still debated. Cells in this region first acquire CD8 and then CD4 at low density. They also rearrange their TCR genes and may express the products of these genes at low density on the cell surface. Maturing cells move deeper into the cortex and adhere to cortical epithelial cells. These epithelial cells are elongated and branched and thus provide a large surface area for contact with thymocytes and the T cell receptors on the thymocytes are exposed to epithelial MSC molecules through these contacts. This leads to positive selection. Those cells which are not selected undergo apoptosis and are phagocytosed by macrophages. There is an increased expression of CD3, TCR, CD4 and CD8 during thymocyte migration from subcapsular region to the deeper cortex. Those TCRs with self-reactivity are now deleted through contact with autoantigens presented by interdigitating cells and macrophages at the corticomedullary corticomedullary junction, a process called negative selection. Medullary epithelial cells might also contribute to this process. Following this stage, cells expressing either CD4 or CD8 appear and exit to the periphery via specialized vessels at the corticomedullary junction. A process of negative selection may also occur in the cortex, leading to the elimination of those cells with TCRs having high affinity for self-MHC. Now, let's study about the development of B cells. The B cells in mammals develop in the bone marrow and fetal liver. Mammals do not have a specific discrete organ for B cell lymphopoiesis. Instead, these cells develop directly from the lymphoid stem cells in the hemopoietic tissue of the fetal liver from 8 to 9 weeks of gestation in humans and by about 14 days in the mouse. Later, the site of B cell production moves from liver to the bone marrow, where it continues through adult life. This is also true of the other hematopoietic lineage, giving rise to erythrocytes, granulocytes, monocytes, and platelets. Recent data have shown that B cell progenitors are also present in the omental tissue of murine and human fetus. Whether or not these B cell progenitors proceed, those in the fetal liver remains to be established. Now let's study about the secondary lymphoid organs and tissues. The secondary lymphoid tissues comprise well organized encapsulated organs, the spleen and lymph nodes, systemic organs, and non-capsulated accumulations of lymphoid tissue. Secondary lymphoid organs can be classified according to the body regions which they defend. The spleen responds predominantly to blood-borne antigens. Lymph nodes mount immune responses to antigens circulating in the lymph entering through the skin or through mucosal surfaces. Lymphoid tissue that is found in association with mucosal surface is called the mucosa associated lymphoid tissue, in short MALT. It is the site of first encounter of the immune cells with antigens entering via mucosal surfaces. Thus, lymphoid tissues are associated with surfaces lining the intestinal tract, that is gut associated lymphoid tissues, in short GALT, and the respiratory tract, that is bronchus associated lymphoid tissue, in short BALT, and genitourinary tract. The major effector mechanism is as mucosal surfaces is secretory IgA antibody, in short SIgA, actively transported via the mucosal epithelial cells to the lumen of the tracts. It is perhaps not surprising that the bulk of the body's lymphoid tissues is found associated with mucosal system, especially the galt, since this is a major pathway of entry for external antigens. Likewise, IgA is the most abundant immunoglobulin in the body. Now, let's study about the spleen. The spleen lies at the upper left quadrant of the abdomen, behind the stomach and close to the diaphragm. The adult spleen is around 13 into 8 centimeter in size and weighs about 180 to 250 gram. Its outer layer consists of the capsule of a capsule of collagenous bundles of fibers which enter the parenchyma of the organ as short trabeculae. These, together with a reticular framework, support two main types of splenic tissue, the white pulp and the red pulp. Now let's study about the white pulp. The white pulp consists of the lymphoid tissue, the bulk of which is arranged around a central arteriole, arteriole to form the periarteriolar lymphoid sheath, in short PALS. Pulse. The pulse are composed of T and B cell areas. The T cells are found around the central arterioles. The B cells may be organized into either the primary unstimulated follicles or the secondary stimulated follicles. The latter possess a germinal center with memory cells. The germinal centers also contain follicular dendritic cells and phagocytic macrophages. Specialized macrophages and a subset Specialized macrophages and subset of slowly circulating B cells which respond to type 2 thymus independent antigens, that is polysaccharides, are found in the marginal zone, the area overlying the mantle of secondary follicles. The marginal zone is only present at one side of the secondary follicle. It also contains NK cells, macrophages, and follicular dendritic cells present antigen to B cells in the spleen. B cells and other lymphocytes are free to leave and enter the pulse via capillary branches of the central arterioles that enter the marginal zone. Now, let's study about the red pulp. This tissue consists of venous sinuses and cellular cords containing macrophages, erythrocytes, platelets, granulocytes, lymphocytes, and numerous plasma cells. In addition to immunological functions, the spleen serves as a reservoir for platelets, erythrocytes, and granulocytes. Aged platelets and erythrocytes are destroyed in the spleen. This process is carried out in the red pulp and is referred to as hemocataresis. Blood enters the tissue via the trabecular arteries, which gives rise to many branches. Central Central arteries surrounded by pulse end with arterial capillaries which open freely into the red pulp cords. Thus, circulating cells reach these cords and become trapped. Aged platelets and erythrocytes are recognized and phagocytosed by macrophages. Blood cells that are not ingested and destroyed can re-enter the blood circulation by squeezing through holes in the discontinuous endothelial wall of the venous sinuses, whereas plasma flows freely through them. The venous sinuses drain blood into the pulp vein and then splenic vein. Now let's study about lymph node and lymphatic system. Lymph nodes frequently occur at branches of the lymphatic vessels. Clusters of lymph nodes are strategically placed in areas such as neck, axillae, groin, mediastinum, and the abdominal cavity, which drain various superficial and deep regions of the body. Lymph nodes protect the skin and mucosal surfaces of the respiratory, digestive, and genitourinary tract. The lymph eventually reaches the thoracic duct which opens into the left subclavian vein and thus back into the circulation. Human lymph nodes are 2 to 10 mm in diameter, are round or kidney shaped, and have an indentation called the hilus where blood vessels enter and leave the node. Human lymph nodes are 2 to 10 mm in diameter, are round or kidney shaped, and have an indentation called the hilus where blood vessels enter and leave the node. Lymph arrives at the lymph node via several afferent lymphatic vessels and leaves the node through one efferent lymphatic vessel at the hilus. A typical lymph node is surrounded by a collagenous capsule. Radial trabeculae, together with reticular fiber, support the various cellular components. The lymph node consists of a B cell area, that is cortex, a T cell area, that is paracortex, and a central medulla consisting of cellular cords containing T cells, B cells, and abundant plasma cells and macrophages. These cells migrating from the skin, that is Langerhans cells, or from mucosae, that means dendritic cells, which transport processes antigens into the lymph nodes from the external and internal surfaces of the body. The bulk of the lymphoid tissue is found in the cortex and paracortex. The paracortex contains specialized post-capillary vessels, high endothelial venules that allow traffic of lymphocytes out of the circulation into the lymph node. The medulla is organized into cords separated by lymph medullary sinuses which drain into a terminal sinus, the origin of the efferent lymphatic vessel. Scavenger phagocytic cells are arranged along the lymph sinuses, especially in the medulla. As the lymph passes across the nodes from the afferent to the efferent lymphatic vessels, particulate antigens are removed by the phagocytic cells and transported into the lymphoid tissue of the lymph node. The cortex contains aggregates of B cells in the form of primary or secondary follicles, whereas T cells are mainly found in the paracortex. Thus, if an area of skin or mucosa is challenged by a T dependent antigen, the lymph nodes draining that particular area show active T cell proliferation in the paracortex. Germinal centers in secondary follicles are seen in antigen stimulated lymph nodes. Germinal centers in secondary follicles are seen in antigen stimulated lymph nodes. These are similar to the germinal centers seen in the B cell areas of the splenic pulse and malt. Germinal centers are surrounded by a mantle of lymphocytes. Mantle zone B cells co-express surface IgM, IgD and CD44. This is taken as evidence that they are virgin and actively recirculating B cells. In most secondary follicles, this thickened mantle or corona is oriented towards the capsule of the node. Secondary follicles contain FDCS, some macrophages and few CD4 plus T cells which interact with the germinal center dendritic cells, and all the cells together with specialized marginal sinus macrophages appear to play a role in generating B cell responses and in particular in the development of B cell memory, which is the primary function of germinal centers. Now let's study about the mucosa associated lymphoid tissue, in short MALT. Aggregates of non-capsulated lymphoid tissue are found especially in the lamina propria and submucosal areas of the gastrointestinal, respiratory and genitourinary tracts. There are three main kinds of tonsils: palatine, pharyngeal, called adenoid when diseased, and lingual, which constitute the Waldeyer ring. The tonsils contain a considerable amount of lymphoid tissue, often with large secondary follicles and intervening T cell zones with high endothelial venules. Similar aggregates of lymphoid tissues are seen lining the bronchi and along the genitourinary tract. The digestive, respiratory, and genitourinary mucosa contains dendritic cells for intake, processing, and transport of antigens of the draining lymph nodes. Lymphoid tissues seen in the lamina propria of the gastrointestinal wall often extend into the submucosa and are found either as solitary nodules or aggregated nodules such as in the appendix. The paired patches are found in the lower ileum, the intestinal epithelium, that means follicle associated epithelium, in short FAE. Overlying the pairs patches is specialized to allow the transport of pathogens into the lymphoid tissue. This particular function is carried out by epithelial cells termed M cells scattered amongst enterocytes and so called because they are numerous macrophores on their luminar surface. M cells contain deep investigations of the basolateral plasma membrane which form pockets containing B and T lymphocytes, dendritic cells and macrophages. Antigens and microorganisms are transcytosed into the pocket and to organized mucosal lymphoid tissue under the epithelium. The deep region of the mucosa contains a cluster of secondary follicles with large germinal centers surrounded by the pair patches. T cell dependent interfollicular zones with interdigitating cells and HEVs. The dome areas is found between the FAE and the follicular area. It contains mainly B cells, the majority of which are memory cells. The intestinal FAE contains M cells, the lymphocytes, and occasional macrophages can be seen in the intracellular pocket. Endocytosed antigens are passed by the root of this pocket into the subepithelial tissues. The intestinal FAE contains M cells and the lymphocytes and occasional macrophages can be seen in the intracellular pocket. Endocytosed antigens are passed by the root of this pocket into the subepithelial tissues. Mucosal, now we'll study about mucosal lymphocytes. In addition to organized lymphoid tissue forming the malt system, a large number of lymphocytes and plasma membranes are found in the connective tissue of the lamina propria and within the epithelial layer of mucosa of the stomach, of the small and large intestine, of the upper and lower respiratory areas, and of several other organs. Lamina propria lymphocytes, in short LPLS, are predominantly activated T cells, but numerous activated B cells and plasma cells are also detected. These plasma cells secrete mainly IgA, which is transported across the epithelial cells and released into the lumen. Now let's study about the lymphocyte traffic. After reaching in the secondary tissues, the lymphocytes do not simply remain there. Many move from one lymphoid organ to another via the blood and lymph. Lymphocytes leave the blood via high endothelial venules. The lymphocytes move through the circulation and enter the lymph nodes and malt via the specialized endothelial cells of the postcapillary venules. They live through the efferent lymphatic vessels and pass through other nodes, finally entering the thoracic duct, which empties into the circulation at the left subclavian vein in humans. Lymphocytes enter the white pulp areas of the spleen in the marginal zones. They pass into the sinusoid of the red pulp and leave via the splenic vein. The movement of the lymphocytes across endothelium is controlled by adhesion molecules and chemokines. Lymphocyte circulation with the mucosal lymphoid system exposes antigens to the to a large number of lymphocytes. Lymphoid cells, which are stimulated by antigen in paired patches, migrate via the regional lymph nodes and thoracic duct into the bloodstream left and hence to the lamina propria of the gut or other mucosal surfaces which might be close to or distant from the site or of priming. Thus, lymphocytes stimulated at one mucosal surface may become distributed selectively throughout the malt system. This is mediated through specific adhesion molecules on the lymphocytes and mucosal HEV. Finally finished. Any questions regarding this? You can ask me.